Let's summarize the basic information you need to know about microarray data. Except where otherwise indicated, the contents of this slide presentation are available for use under the Creative Commons Attribution 4.0 license. You are free to adapt and share the work, but you must give appropriate credit, provide a link to the license, and indicate if changes were made. Microarrays are a fast and cost-effective way to survey the whole genome at once. Although they are less commonly used since the rise of lower-cost whole genome sequencing, they may still be a suitable choice for your genomic study. There are multiple types of microarrays, but they all involve hybridizing sample fragments to a chip with fluorescently labeled probes. The amount of fluorescence is determined by analyzing an image taken of the chip. Some microarrays are designed to work with DNA samples. These microarrays detect the presence of particular SNPs or variants within the genome. When the sample is hybridized, the amount of fluorescence detected can be interpreted to indicate the presence of the variant and whether the individual is homozygous or heterozygous. Other microarrays are designed to work with RNA samples. For these microarrays, RNA samples first undergo reverse transcription, then are hybridized to the microarray chip. The higher the fluorescence, the more RNA that is being detected in the sample. These particular microarrays are useful for determining gene expression levels in a sample. Finally, microarrays can also be used to survey DNA methylation. DNA samples are prepped using bisulfite conversion to convert unmethylated cytosines into uracils. The methylated cytosines are left untouched. Probes bind to either the uracil or the cytosine. A ratio of the fluorescence signal can be used to identify the relative abundance of the methylated and unmethylated versions of the sequence. When preparing to analyze microarray data, you should make sure you get details of the lab process before you begin. This includes the experimental design, what the microarray is measuring, and whether the microarray is 1 or 2 color. You will also want to know details about the chip itself, like what brand of microarray chip it is, and what is the specific name of the chip design. Both of these details can change how you go about doing your analysis. Finally, make sure to ask if any pre-processing has been done to the data and what those steps were. This includes finding out what normalization method may have been applied.